Okay, hi classmates. I'm Sungdong Lee from Protein Laboratory. I'm supervised by Professor Sang-Yeol Lee, and now I'm in third semester of PhD course. Today, I'm gonna present about modification of signal pathway in plants through alternative expression of protein and its application. In the cell, basically, when a signal pathway is transduced through several steps like this, with typical research, if you expect to enhancing some genes expression, the easiest way is overexpressing the genes or that transcription factor, but in many cases, there are various unexpected side effects of direct overexpressing the genes or their transcription factors. In that point, I'm studying about modifying the signal pathway with more specific way without or minimize the side effects which we don't expect. I prepare the presentation about my previous research as an example for that. As you well know, plants are sessile organisms and they face very diverse abiotic environmental cues like light, drought, and temperature changes or ionic stress containing heavy metal or biotic attacks from pathogens and insects. So plants have to evolve some resistance maneuver with signal pathway against them. Especially the temperature is a major environmental factor which restricts the plants to make limitation for their geographical distribution. Among them, I studied about the low temperature, the cold stress. The chilling or freezing temperature work as very harmful abiotic stresses to plants. It affects the growth and development of plants, especially to crop plants. It is very serious problem triggering the reduce reduce of merchantability. But for a less long time, plants evolved to gain some signal pathway to show a tolerance that against the cold stress, and is it was very well studied by many researchers in several years. In simple, when cold stress when a cold stress is sensed by plants, some physiological some physiological damages like growth inhibition, plasma membrane disintegration, metabolites imbalance, etc. are occurred. But within some signal is transduced, transcription factors are activated and cold responsive genes are expressed to resist the stress. In this pathway, there is a key transcription factor, CBF. The CBF works as a most major transcription factor of cold responsive pathway. As as expected easily, the CBF overexpressing plants show more tolerance show more tolerance to with cold stress and the knockout mutant of that shows very sensitive against low temperature treatment. But there is very serious problem with enhancing cold tolerance of plants with just overexpressing the CBF. The transgenic plants with overexpressing CBF shows defective phenotype under normal growth and developmental conditions. So, even if the plants obtain increased low temperature resistance, it is not available because of the retardation of basic growth. To sum up, in normal growth condition without the cold treatments or cold stresses comes, CBF protein is needed to grow and develop without the turning on the cold responsive genes, and under normal condition, under cold stress condition, it is also needed to induce the expression of cold responsive genes. In that point, I assume that maybe there is a regulation mode of activity of CBF transcription factor protein. According to several research, there are so many reported studies about regulation of CBF transcription factors expression, but all of them are just about expressional regulation of CBF, not about activity of that on protein level. I set a hypothesis about there is a regulation mechanism of activity on protein level with post-translational modification. The post-translational modification is a way of regulation of protein's physiological function with phosphorylation, methylation, or forming a disulfide bond, hydroxylation, etc. The PTM is occurred by enzymatic protein, and when the PTM is happened, there is also protein-protein interaction is occurred. So, I got a curiosity of which protein is interacting with CBF transcription factor and motivates its PTM. To solve that question, I screened the interaction partner protein of CBF transcription factor, and I found the thioredoxin H2, which is a protein has an enzymatic activity in cell. Thioredoxin protein regulates target protein's redox status from oxidized to reduced. Through this function, thioredoxin protein mediates very diverse regulation of subcellular physiologies like apoptosis or apoptosis, DNA replication, transcription factors activity change, cell division, and etc. In previous, thyroidoxin mediated redox status change motivates target protein structure change was reported, and I assume in CBF case could be very similar with previous reported. So I got a question with, does redox regulation of CBF transcription factor by thioredoxin H2 lead to its structural change? After several experiments, I could confirm the thioredoxin H2 mediated structural changes of CBF proteins, and this structural change confers activity change of CBF transcription factors. With those results, I could conclude each condition of non or cold stressed condition, thyrodoxin H2, the TRXH2 mediated structural changes of CBF protein can regulate the cold stress tolerance in plants with expressing the regulation of expressing the cold responsive genes. With the conclusion of the project, I thought this regulation module could be applied to crop plants, especially points of improvement in storage of crops. So I wondered the introduction of TRXH2 genes into crop plants, enhancing the cold tolerance or improve the storage ability at low temperature of crop plants. I introduced the TRXH2 genes into sweet potato and checked some physiological phenotype with low temperature treatment, comparing with non-transgenic one. TRXH2 transgenic plants show very tolerant phenotype to the cold treatment and even shows much more improvement with efficiency of biosynthesis. Likewise, firmness of transgenic plants was tested, and transgenic was showed much stronger than non-transgenic after low temperature storage condition. So the application of TRXH2 genes to the crop plant enabled to tolerate under cold stresses, and now I try to test it even in another crop plant like cabbage or flowering plants. Like this study, I tried to analyze the signal pathway and make alternation of the regulatory protein of the pathway, expecting the enhancing physiological ability of plants with minimizing unexpected side effects. With these kinds of studies can make me to be a good researcher, and these kinds of studies can be a good training and options for me to be a good researcher in field of not only in plants over the cellular biochemical research field. Thank you for listening.